everybody. Welcome back. We're going to get into the different chemicals that control microbial growth. So when you're looking at specific chemicals or types of chemicals, you have to look at the toxic effects of the chemicals. So the benefits are going to have to outweigh the risk of use, because if the chemical is more toxic, if it is a known carcinogen, then you're not going to want to use it. But if the chemical maybe just has a fume that is uncomfortable to smell, then that might be still beneficial to use that chemical if it's going to get rid of certain microbes. The activity, so how does it work? Does it work in the presence of organic material, or is it inactivated by organic material? Because that's going to determine what type of chemical you're going to use. If you're using a chemical to help you clean wounds, you need it to be able to work in the presence of organic material. Compatibility with what you're using or what you're treating. So you can't use liquids on electrical equipment, it'll ruin them. What type of residue is being left over? Is the residue going to corrode whatever you're cleaning, or is it toxic to the environment or to you? The cost. So you always have to look at how much is it going to cost, and will it be available for you to use? If it can't be made, if it takes 24 days to make some chemical, then it probably isn't worth making the chemical unless you're going to have a huge system that is making it continuously so that you have enough. And then if it costs too much, then it's not worth it either. And then how are we storing this chemical? If we store it in a bottle, is the bottle going to corrode over time? Is it stable in that bottle? So you want to make sure that whatever you're storing it in is going to be safe. And then the environmental risk. When you dispose of the bottle that has the residue of the chemical, is it going to cause problems? Do you have to do anything before you dispose of the container? If the material gets into water supplies, what's going to happen? These are all things that we have to take into account when we're choosing the proper chemicals to control growth. So we have different tests we can use. And so the metal cylinder test, you're going to take these cylinders and you dip them into a broth that has bacteria. So you leave them in the broth, then you take them out and you let them dry. Once they are dry, you then dip them into a chemical, some dilution of a chemical disinfectant for 10 minutes. Remove them from the chemical disinfectant, wash and place that metal tube, or wash and place that metal cylinder into a medium of some sort, and then let it incubate for 48 hours. So if the medium, after that 48 hours, if there's no growth, then that tells you that that chemical was effective at getting rid of the microbes. So if it's at a high dilution, that means that there's very little chemical. If you have 100% chemical, that's 100% of the solution. That would be not diluted at all, right? But if you have no growth and there's only, like, say 5% solution, so 5% of the chemical in 95% of water, and it's still effective, that tells you that this chemical is really good at killing off whatever microbe that you're looking at. Another mechanism to test multiple chemicals is known as a disc diffusion test. So in this test, you're going to take a medium of either tryptic soy agar, or you can use Mueller-Hinton agar if you're testing antibiotics. You then take discs that are sterile and you dip them into the chemicals that you're trying to test, right? You make a lawn of bacteria over the TSA plate. You take the discs that are in the different chemicals and you tamp them onto the plate. So you just kind of push them down with a sterile set of tweezers or forceps. And then you invert and incubate them for about 48 hours. Once you're done, you then measure the zone of inhibition. So here we have a zone of inhibition. If we were to measure it, I don't have a ruler on me, but if we were to measure this zone here from the middle here out, and this one here, obviously this one has a larger zone, right? This one has a very small zone. This one has almost no zone of inhibition. And so that's going to tell you which chemical is best at controlling microbial growth of whatever you made a lawn of. So if this was E. coli, then you know that this is pretty effective, this is effective, these two are pretty effective at killing off whatever this is. If you want to know what's best at killing off, say, staph, you can use this same test. You'd have to make a plate of staph and then run the test with staph on there, because E. coli and staph are very different, and so they're going to have different results. And so here's a question for you that I'll have you guys figure out, and I'll ask this. So I'm asking you this. I'm not going to give you the answer, but if you have questions, you can ask me, okay? So if you're trying to clean a wound on your child, assuming you have a child, which one of these agents would you use to kill the wound or to clean the wound? And then the next question is, what if you're trying to clean your countertop? Which agent would you use to clean your countertop? You can think about that, and I'm going to go to the next slide. So now let's look at some chemicals that are used to control microbial growth. The first type of chemical are heavy metals. So in the past, we've used heavy metals to control microbial growth. And they do. They can interfere with protein synthesis, or interfere with proteins. And so if proteins are not functioning properly, then you cannot replicate cells. They are used for disinfection, and they've also been used in preserving materials. Silver nitrate is still sometimes used to treat the eyes of newborns to prevent blindness. In a lot of places now we have moved from silver nitrate to something like erythromycin. But silver nitrate has problems or has drawbacks. And so when you have a buildup of silver in your skin, it causes a problem known as Argyria. And so here you can see a normal hand right here. And this is a hand of an individual with Argyria. This is the face of an individual with Argyria. So this is an irreversible condition caused by accumulation of silver in the skin, and so you have this purpley blue color to your body. The next chemicals for controlling microbial growth are called halogens. Halogens include chlorine, fluorine, and iodine. These are used often for either disinfection or for treating water supplies. They function by denaturing proteins. And so we tend to use chlorine to kill pathogens found in water, and because chlorine doesn't have a horrible aftertaste, the water that we typically drink is chlorinated water. Not a lot of chlorine, but there is some chlorine in it. So chlorine in pools is not the same. That's a lot of chlorine, and too much chlorine can cause inflammation of the conjunctiva and cause itchiness of the eyes and can cause itchiness on your skin as well. Iodine has been used for a long time as an antiseptic. So it is used to clean wounds and to treat skin prior to surgery. So what they typically do is they complex iodine with another molecule. It's called an organic carrier. And what that does is it slows the release of iodine, because iodine in and of itself can cause burns. Especially if you have an autoimmune response to the iodine, then your skin's going to start burning and it'll turn red and it'll be painful. So betadine is an example of an iodine complex to an organic molecule carrier. The next chemical is alcohol. So alcohol is used to, alcohol denatures proteins, and it also disrupts the plasma membrane. And it can be used as an antiseptic as well. But alcohol does evaporate very quickly. So typically when you're using alcohol, you're going to have to use it quickly and you're going to have to use a lot. So phenolics are more stable and they last longer. They also denature proteins and disrupt plasma membranes. But since they are more stable, they don't evaporate so fast. They are more common in your household. So they're used in household cleaners. They're used in disinfectants. They're used in mouthwashes. They're used in a lot of stuff. So let's look at some ways that we use phenols. Triclosan is a phenol. It's found in antibacterial soaps, in plastics, in textiles. It is an antibiotic in and of itself because of how it inhibits bacterial fatty acid synthesis. So you can find it in toothpastes, in cosmetics, school supplies, just everything to control microbial growth. Then we have bisphenols, so another type of phenol, and Lysol, which is one of the most commonly used disinfectants. Lysol kills cold and flu viruses. So when you say kills, it inactivates them because viruses are non-living, but it does get rid of them. So what it does is it disrupts the cell membrane. If you are a virus and you have a protein or a coating around you, then it can disrupt that envelope and inactivate the virus that way. Not protein, I'm sorry, I said protein coat, but I meant it's a phospholipid envelope that surrounds some viruses. And this is what it inactivates, or it stops that synthesis. And so the cells or the viruses can't function. Actually, the coronavirus has an envelope around it. So Lysol will actually inactivate the coronavirus, which is what everybody is concerned about these days. Surfactants. So surfactants are materials like soap or detergent that decrease surface tension. So they basically help to mechanically carry microbes away. Water is great. Water, using water is better than using nothing when you're scrubbing your hands. But when you use a surfactant like soap, then you're actually producing an emulsifier that helps to pick up the microbes even easier. So some types of surfactants are called cationic surfactants. And so quats are, or quaternary ammonia compounds, which we typically just call quats, are cationic. And so they are attracted to the cell wall of your bacteria. And when they bind, they actually will start breaking down the cell membrane. And so they can help in actually killing the pathogens as well. Not just removing, mechanical removal, but actually killing also. Alkylating agents then are your, or are chemicals that contain groups known as aldehydes. And so formaldehyde, glutaraldehyde, orthophthaledehyde, are all types of alkylating agents. They have alkyl groups, okay? But alkylating agents are carcinogenic, and they also cause irritation to skin. They function, they sterilize the environment, they inactivate proteins and nucleic acids. Typically, we use aldehydes in storage of tissue specimens. We use aldehydes to embalm organisms. And we also use some aldehydes in vaccine preparation to inactivate infectious agents. So I always like to remind you that there's always exceptions. And one exception is this very, very strong egg that is microscopic. This is the egg of Ascaris lumbricoides. Ascaris lumbricoides causes ascariasis, which are these large roundworms, roundworm infection, which when a child or when someone is infected with these roundworms, if they have a very, very severe infection, the worms will actually move through their body. They will come, they can cause they can block, that's it, bowel blockage. But they will also move. If they're in an area where they're not getting enough nutrients, they're going to move around, and your GI tract is one long tube, so they're going to move, and they might move up and out the mouth or the nasal cavity. And so this is an individual that has a very serious Ascarias infection. And why am I showing you this? Because these eggs can withstand the aldehyde. So if you are ever doing any dissection of Ascaris worms, the eggs are still viable. If they get into your mouth, they will move into your gut and hatch, and that can be dangerous. One Ascaris female can carry millions of worms, worm eggs. Then we have ethylene oxide. Ethylene oxide is also a sterilant. It is a gas sterilant, but it is also carcinogenic and it is explosive. So it can permeate through heat-sensitive packaged materials. So what we typically use is we use an ethylene oxide chamber. We put materials that can't be sterilized in other ways into this chamber, and then we turn it on, let it run, and the gas sterilant will kill microbes in here. Then when you open it, the material that's inside is going to be sterile without any fluid of any sort, okay, or even without increasing temperature. So like if you have shoes that you wear at work and you need to sterilize them before you go home, this would be an example of why you might do that. All right, that's the end of our chat. Oh, I lied. I have one more. Sorry. The last one, this is the end of our chapter here, is peroxygens. Peroxygens are used to kill bacteria, and they function by oxidizing the material. So basically, they oxidize the cell wall, cell membrane, and produce free radical, and then in doing so, they produce free radicals. Okay. So peroxygens include your hydrogen peroxide, benzoyl peroxide, peracetic acid. These are highly effective disinfectants, and so they're often used in cleaning things like contact lenses, or they might be used to help in cleaning your mouth when you have an oral wound. So hydrogen peroxide for that one. I don't know peracetic acid at all, actually, but this is an example of hydrogen peroxide cleaning a contact lens. And that's it. So we are done with this chapter. I will post these videos shortly, and you guys have a wonderful day. Bye.